And so we were looking at this idea that the first enzyme in the C3 cycle, rubisco, can take either carbon dioxide or oxygen into its active site. And if you have the CO2 coming in, then you run the C3 cycle, which is being shown here in the big picture kind of pie chart image of it, where you see the carbon dioxide coming in at the top right, and it is going to react with RUBP in the active site of rubisco, the rubisco enzyme. So if that happens, then great, you have this C3 cycle, and you have a way for the plant to make either sucrose or starch. It can keep running this cycle to make sure it has enough RUBP coming, being regenerated, so that CO2 coming in can react with it. So that's all great. But we have talked about how sometimes the plant will be put into a situation where the rubisco enzyme does not see enough of the carbon dioxide coming in. Instead, it's oxygen. And then rubisco's like, fine, I'll take that one in too. So whatever is more abundant around rubisco, that's what wins and comes into the active site. And you get this big complicated PCO cycle kicking in. So these are both the depictions of the PCO cycle. The left one's more detailed. The right one is a little bit pared down, but basically you see it's complicated. You got 3 different organelles. You have a big long pathway to look at. You have some issues in the sense that you have a loss of carbon in the form of carbon dioxide. Okay, so they're showing that in both images there. So that's just respired off. So that's why they call it photorespiration. And there's extra energy expended. Here you can see ATP is being used to do the do the last step of taking glycerate and making phosphoglycerate off of that. But once phosphoglycerate is made, that's great, because then it can go right into the Kelvin cycle and continue the Kelvin cycle. So that's good. But still, that's a lot of effort to just and essentially they go through this twice to make one PGA molecule that they can push into the C3 cycle. They've lost carbon in the form of carbon dioxide. So I think we left off around there where we're looking at that there is a cost, but they can recover 75%. And then the idea is, okay, why would rubisco be sloppy like this? What's going on? And then like, under what conditions do we see oxygen winning and coming into the active site? So when you look at, they've done studies on this, of course, and they've looked experimentally, and they know that when you look at like roughly room temperature conditions, 25 degrees Celsius, and you have both carbon dioxide and oxygen dissolved in liquid, like in a cytoplasm of a plant cell, you have a ratio of 3 to 1. That means you have 3 times as many carbon dioxide molecules in the solution compared to oxygen. That's good. That means rubisco will see lots of carbon dioxide. But as you know, a lot of plants don't live at 25 degrees Celsius year round or even within one day. And so if there's a condition that forces more oxygen to be, that ratio shifts, so you get more oxygen instead of CO2, then, whoops, that's the next bullet, then oxygenation is favored, okay? But if you create a situation where there's more CO2 than that second bullet point, if there's more CO2, you get carboxylation favored, all right? So it's this idea that, okay, as long as you can keep that 3:1 ratio, that's good. But there are going to be conditions where that shifts to the point where oxygen wins and there's more oxygen. And so if you think about it, if you're a plant and you're growing vigorously in the summertime, let's say it's, June, late June, and your roots are going, hey, I don't have a lot of water in the soil to draw from. I'm starting to experience drought. What could you do as a plant to respond to this? I'm not getting enough water in. What might you do to another part of your body? what might you do? Yeah, close your stomata, right? Because you don't want to lose any more water out to your body, out to the atmosphere, the water vapor. So you'll go ahead and pull down your stomata. Okay, so you're doing that, and that's a good solution temporarily to this idea that I'm not drawing in enough water. Okay, so if you close your stomata, that means you don't have any new carbon dioxide coming in. You have some still left, you know, if it's high noon, you've been busy all day bringing in CO2, but then you close down your stomata. Now your CO2 inside your cells, it will get used for photosynthesis, but those levels will plummet, right? They're going to drop. And if your stomata stays closed, you're not bringing in more. So now you've shifted it. You created a situation where now oxygen is still available in your cells all around. All right. So now you've tipped the ratio in favor of oxygen. And so if you're the type of plant that experiences drought, you're going to have an issue. You're going to have Rubisco seeing too much oxygen. If you have increased temperatures, which also tends to go hand in hand with drought typically, so increased temps, increased drought conditions, those will favor oxygenation. And so the other thing too to realize that as the plant cells start to warm up, what happens is carbon dioxide, it becomes less soluble. It kind of like bubbles out of solution when it gets hotter, but oxygen doesn't do it as quickly. So that means oxygen sticks around longer in cytoplasm, and of course can then get pushed into the PCO cycle. You have Rubisco taking oxygen. So you have that issue too. With increased temperatures, you just get this shift in solubility. So that's a problem. And then the drought, they're also finding salinity. There's plenty of soils out there that are saline, whether it's because they've been over fertilized in commercial ag fields, or there's just some plots of land that are naturally saline. And so again, that along with drought promotes this idea of Rubisco seeing oxygen over carbon dioxide. And then we talked about this last week, just the idea that Rubisco evolved on this planet when the planet didn't really have oxygen in the atmosphere. So back then, Rubisco never had to worry about oxygen, so to speak. And so it's understandable why Rubisco never really cared about oxygen, because it didn't even know it existed, so to speak. But, you know, fast forward 2 billion years later, and it's still doing that, and it still doesn't care really if it sees the carbon dioxide or the oxygen. So it really hasn't tightened up, and that's still a puzzle to scientists studying it. And the other plausible hypothesis of why Rubisco might not really, why it takes both, is that sometimes the PCO cycle could be beneficial in the sense of some of the intermediates that get produced. Perhaps those are important at key moments in the plant's life where those come in handy. And that's certainly possible. But again, I think I told you last week that if you run plants, if you force them to run the PCO cycle consistently, like a lot, they suffer for it. And so in general, the idea is that this is okay some of the time, you just don't want to plant doing this a lot. And so, but there could be conditions that a plant sees consistently where it would force PCO. So hot temperatures, like we were just thinking about, dry soil, all of that could create problems. So that's where they started looking at these plants that live in consistently dry, warm conditions. How are they dealing with this? How come they're surviving okay, doing quite well? Are they running the PCO cycle? So they went looking for that, and they found that there are groups of plants that don't seem to really run the PCO cycle, or hardly at all if they do run it. And so that's the next slide set that I have posted into EDUCAT on the adaptations that some plants have made in order to, I'm going to stop share and reshare, in order to make sure essentially that you have Rubisco continuing to see more carbon dioxide than oxygen. So let me reshare. And so this one. So we're looking at the idea of these two cycles. So the C4 cycle and the CAN cycle or the CAN pathway. So these are some physiological adaptations that have been done to deal with rubisco being sloppy and being able to take in oxygen as well. So the C4 cycle, you're seeing a jungle, tropical jungle, and so that's what we're thinking about. We're thinking about plants that live in and are native to tropical or subtropical conditions where you have consistently warm conditions and periods of drought, right? So now if you look at the light reactions and what we were thinking about in that process, you have the light reactions feeding in. You still remember, I'm just putting this in here to remind you, you have the NADPH and the ATP, and we know that those are playing a role in the C3 cycle. So now we're going to see that the C4 cycle also does play a role in that the plant has to expend extra energy in order to get the C4 cycle to occur. So I'm going to go into this. So when you look at C4 plants, they essentially tack on this so-called C4 cycle and kind of link it to the C3 cycle. But it's not done for free. There's a cost, an extra ATP cost. And so when they looked at these plants and studied them more carefully, like how are they doing so well? How are they minimizing the PCO cycle? What they discovered is when they look in the leaf, they would see an obvious difference in leaf anatomy. I'll show you some pictures to prove to you what I'm talking about. So we're going to zoom in these pictures to the bundle sheath and the mesophyll or parenchyma cells right next to the bundle sheath. And I'm going to show you that there is this spatial segregation of where these reaction pathways happen. So we're going to see that the C4 cycle occurs in one physical spot in the leaf, and then the C3 cycle in a different spot. And so we say they're spatially segregated, but that turns out to be a really good thing, and I'll show you what I mean. So yes, every single plant has a C3 cycle. We have yet to find a plant that can't do that. And so the C3 cycle evolved first. That was on the scene first in the process of evolution. But then some plants started playing around a little bit and evolved the C4 pathway to deal with the fact that they're always in pretty warm conditions. And so when you study these plants that do this, they can handle drought better than just straight C3 plants. They can do hot temps better than C3 plants and water stress better. And so they outperform C3 plants in these types of environmental conditions. And when they study them in the lab, they show that they can have, C4 plants can have 2 to 3 times the photosynthetic rate compared to C3 plants. And one of the huge payoffs to running the C4 cycle is this last bullet point, where you have essentially, when you look at what's going on, what it's really doing is it's concentrating a bunch of CO2 right around rubisco. So it's creating a situation where rubisco is just, I like to think of it, rubisco taking a big bubble bath, where all the bubbles are this lovely CO2 that's just bathing the rubisco. And so then if you create that situation, you've essentially eliminated the PCO cycle. So I'll work through some of the imagery here now to show you the leaf anatomy, and then we'll get into a little bit of the actual pathway. So here's a comparison of, well, these are both C4 actually. These are C4, two different images. So what are we talking about? So this so-called Kranz anatomy, this word that you see up top in figure A, Kranz, that means wreath in German. And so they're talking about the bundle sheath cells that form a ring or a wreath-like circle around the vascular tissue. So inside of that is the vascular tissue. So there's the bundle sheath here in the big central vein. The bundle sheath is encircling that. Here again, here's the bundle sheath. And especially with the minor veins, you can really see how plumped up those bundle sheath cells are. They're really big. And they tend to be chock full of chloroplasts, lots and lots of chloroplasts. So that was one of the first things they noted, like, wow, that's different. C3 plants don't have that. They have a bundle sheath, but they're really average in size. Nothing, it doesn't like pop you in the face with, wow, look how big those are, okay? And then here's just another image, excuse me, trying to show the large bundle sheath cells, lots of chloroplasts. They stained it a little differently, but the blue are the chloroplasts. Okay, so those are the veins of the leaf. The bundle sheath surrounds the vein, the vascular tissue. And the other idea, I think I'll switch to a drawing. It's a little bit easier to see. The other thing too is in addition to up top is your C4 plant. So here they're trying to show that the bundle sheath in blue here are beefed up in size, they're plump. And then they're trying to show how some of the parenchyma cells or mesophyll cells hug up to the bundle sheath, like they're right plastered next to them. But if you look at a C3 plant, that doesn't really happen. You see certainly a bunch of parenchyma cells in there, but they're more like just hanging out in the inner tissue of the leaf. They're not smacked up against the bundle sheath. So that's an obvious difference between C4 and C3. And so and then when you look at these parenchyma cells that are hugged up to the bundle sheath, when you look at that more carefully, you see that there's all kinds of plasmodesmata, those tunnel connections between the bundle sheath and the parenchyma cells. And that's important because if we go to this, what they're trying to show here is the plasmodesmata. The plasmodesmata are needed in order to link the two cycles together, the C3 cycle and the C4 cycle, because you have to get this movement of key molecules that will allow for the linkage of these two cycles to occur. So what we have happening, this is kind of like the pie chart idea where they're breaking it down into three different sections of the C4 cycle. So what they're showing here is in a mesophyll or parenchyma cell, you have, let's say it's daytime, stomata are open, CO2 is being brought in. And what you see is that in these C4 plants, they don't react to CO2 with rubisco right away. They don't do that. Instead, they use a different enzyme and it goes through a carboxylation. So I'll explain about that. So let's go to that right now. So what they're showing here, what the carboxylation is being shown here in more detail. Here's the CO2 up top coming in, and you see that it's the enzyme PEP carboxylase. It's not rubisco that immediately works on that carbon dioxide. So what it does is this PEP carboxylase takes in phosphoenolpyruvate, PEP, and then CO2, and reacts those together, and you get this oxaloacetate molecule. So that's why they call it a C4 cycle, because oxaloacetate has 4 carbons. It's a 4-carbon product. Then you can see that oxaloacetate goes to malate through this small reducing step here, using NADPH from the light reactions. So that also is being used. Okay, so then you get malate. So then I like to think of malate as a vector. When you think about how we describe vectors in biology, they're some kind of object or organism that can transfer something from point A to point B. The classic example is malaria, how the mosquito is a vector for the malaria pathogen. And so what I like to think is malate is a vector for carbon dioxide, because essentially carbon dioxide is part of its body, part of its chemical structure. And so now it's easier for carbon dioxide to get mobilized between cells if it's part of a structure rather than just dissolved gas. That's much slower. It's not so precise. So here you have malate essentially carrying carbon dioxide as part of its structure. Malate will mobilize out of the mesophyll cell and get into the bundle sheath. And then it delivers the CO2. It goes right to where rubisco is, cleaves off the CO2 and says, here you go, rubisco, I'm giving you a bunch of CO2. So if you can imagine this happening over and over and over again, you can see how Rubisco is getting bathed in CO2 by the help of malate, delivering it from the mesophyll to the bundle sheath. So this, excuse me, this delivering of the CO2 and cleaving that off, that's the decarboxylation. So the C4 acid that they're showing on this slide, that's malate. That's malate coming in to the bundle sheath and cleaving off the CO2 and giving it right to Rubisco. So you can see how the artist drew this. Calvin cycle, the word Calvin cycle is showing up in the bundle sheath, but you don't see it showing up in the mesophyll. So they've spatially segregated where they allow the Calvin cycle to happen. And that's a good strategy because that makes sure they've created this situation where malate moving into the bundle sheath cell where Rubisco is going to bathe Rubisco. And so it's a very precise way to make sure Rubisco's seeing a bunch of CO2. Okay, so carboxylation, that's bringing in the CO2 and reacting it with PEP, with that very nice enzyme, PEP carboxylate. Decarboxylation is malate delivering the CO2, getting it to Rubisco, so the Calvin cycle can run. Well, when you cleave off CO2 off malate, you're left with another product, which is pyruvate, which is being shown here. Okay, so pyruvate is left over. So the plant says, okay, let's push that back over into the mesophyll cell because we can build phosphoenolpyruvate off of pyruvate. So let's put that back in the mesophyll cell, and with this extra help of ATP, it will convert pyruvate to phosphoenolpyruvate. So that is regenerating the phosphoenolpyruvate, all right? And so that way the C4 cycle can keep happening all day long. As long as you have enough phosphoenolpyruvate ready to go, then the CO2 coming in has something to react with. And so what you end up doing is creating a situation where the Calvin cycle cooking along in the bundle sheath is spatially segregated in the bundle sheath. It's seeing a ton of CO2 coming from malate. And what we also see, and that's not being shown here, is that the light reactions of photosynthesis aren't really happening in the bundle sheath. They also kind of split that away. They're like, you know what? I don't want the light reactions going on in the bundle sheath, because when you think about the light reactions of photosynthesis, we tend to focus on, well, you get NADPH, you get ATP, but there's another product created during the light reaction that we don't currently focus on. We certainly need it. What is it? What are we sucking in right now? Oxygen, right? So the light reaction, one of the products is oxygen. If you have light reactions going on in the bundle sheath where the Calvin cycle is, that's not good. You have oxygen now right around the Rubisco. So these C4 plants, they sequester that too. They say, okay, the bundle sheath, let's just do the Calvin cycle. Let's put the light reactions off into the mesophyll cells instead. And so that's another spatial segregation that they're playing in order to keep Rubisco from not seeing the too much oxygen. Questions on the C4 cycle or mechanism? Yeah. Yeah. So I think when you're learning this, if you could focus on making sure you see the bigger picture here, like this is like the bigger picture idea where you have the three different distinct events. You have carboxylation, decarboxylation, and then regeneration. So, and then we're kind of zooming in here on this slide into more detail. So what you're seeing here is once the malate is getting made, it gets mobilized. It's a vector. It moves from point A to point B, out of the mesophyll into the bundle sheath. And then the CO2 will come cleaved off of it. Okay, so you can think of maybe this one. And that CO2 will get cleaved off and fed to Rubisco. And so if you do that, if you take a CO2 off of malate, then you're left with pyruvate. Yeah, it does. So this is a good thing, right? You have NADPH being created here, which is good. So even though, so when they talk about this cycle, the C4 cycle, they don't really say that there's a net cost of NADPH because it's used here but made here. So that's nice. But they do say there's a net cost of ATP. So nowhere in here do you see ATP being created. It's only getting used. So when they talk about the cost of the C4 cycle, they zoom into, yeah, you gotta use some extra ATP. And it's a really funky enzyme that they've been studying. I'm not gonna get into that, but it's a really unusual enzyme that runs this reaction. So the next series of slides are just in Word. I like printed out in words what we are just covered. I'm not going to go over that right now. That's for study purposes. But it depends how your brain works. If you'd rather see the visual, if you'd rather see the words, I gave you both to focus on and look at. So we use what works for you. Here's the overall cost. So they say that per CO2 molecule getting fixed through this C4 cycle, you have a net, two extra ATP get expended. And so if you combine that with the cost of the C3 cycle, because the C3 cycle uses ATP, it would be a total of 5 ATP and 2 NADPH to run the C4 and C3 combined. If you just zoom in on the C4, then it's 2 extra. But if you combine the C4 and the C3, it's 5 ATP, 2 NADPH. So that means this cycle is not free, energetically speaking. There's a cost. But when they look at the payoff, it's worth it. It's worth it for these plants because they live in such consistently hot conditions, pretty warm conditions all year round. And then they go through periods of drought. If you want to tropical forest, there's droughts. And so these plants have evolved over time, the C4 cycle. And, so of course they're really interested in, this is a really cool spin-off and a really cool way to deal with this situation. So they started looking like, who else can do this? Like, is this like just a certain family of plants? Is it, like, who can do this? And so what they've been finding so far is that there's about 19 different families of angiosperms. And again, it can be monocots, it can be dicots, that doesn't seem to matter. The grasses are a huge one. There's a lot of different species of the family Poaceae that can do this. But also the Asteraceae family, which is one of the largest families out there, and many others, so 17 others in addition to those. And so about 3% of all known angiosperms can do the C4. You think about some of the more commercial important ones, corn, maize or corn, sugarcane, sorghum, millet, and some sedges can run the C4. And so, if you just think about all the corn being produced worldwide, and they are running the C4. So, we have cornfields up here in the UP. We are not tropical, right? So it kind of begs the question, how efficient is it to grow corn up here when they're C4 plants and they are spending extra heat to do that, but they're in an environment where they don't need most of the time. So, that's something I think that's worth thinking about when you grow crops. Like, where do you grow them geographically to make the most sense and make the most profitability? So, if you grow corn here, they're running the C4, but they don't really need it most of the time. And so that's kind of a disadvantage to them in this environment. Sugarcane, of course, we don't grow that. It doesn't seem to be capable of growing up here, but again, that's probably $1 billion industry worldwide as well. So they are definitely looking at, can we take the C4 cycle and put it into other plants that are commercially important? And they're working on it. I'll talk a little bit more about that. Unfortunately, there's some very aggressive weeds that are also considered or capable of doing the C4, like crabgrass, Russian thistle, pigweed. So some of these show up in your lawn. And so people who want this beautiful monoculture lawn of the nice grass, but you get these other weeds showing up, it can be very challenging because they can outcompete those straight C3 plants in certain geographical regions, they can really thrive. So they've certainly looked at the evolution of the C4 cycle, and they 70 different independent times it's evolved. That's a lot. So it's kind of fun to think about how the plants have been. I like to think of them like playing around. Does this work? How does this work? If I tweak this, if I mutate that. And so 70 different times, at least, that this has been done in the evolutionary process of creating the C4 cycle. And so it's not like it's the single point in time where, boom, you have the C4 and then everything radiated from that point in terms of who can do it. And it's not that straightforward. So C4 photosynthesis, 40% greater ability to convert sunshine into biomass than C3. And maximum growth rates and yields of the most productive C4 species are 40 to 50% greater than those of the most productive C3. So they grow faster. They can create a lot of biomass if you run them head to head with C3 plants. And so that last quote was taken from a paper. When was that published? A while ago. And so you have, that there's a good interest in, can we get other plants to do this? Because they see how well these plants can grow under conditions that are more close to the equator, where you have plants being grown for production in those areas. And so, for example, the Rice Project, the C4 Rice Project, is a global initiative where they're trying to create C4 rice. So C4 rice are C3 plants. And the question is, can we create rice plants that run the C4? And they're making progress, but it's not quite there yet. As you can imagine, it's incredibly complicated to engineer this pathway in the plants. That's a lot of tinkering. And they're approaching it through a lot of synthetic biology, molecular biology methods. And like I said, it's a global initiative. This link down at the bottom here, this YouTube link, would get you to see this short little 7-minute interview with one of the main founders of the C4 Rice Project. And so I'd like you to look at that in your off time. But it is one avenue they're pursuing to think about how do we feed all these billions of people, and our population keep increasing. We're running out of land. How do we do this? Well, this is one thought. Could we ramp up the efficiency of our crop plants by engineering a C4? And I think within your lifetime, you'll see this happen. Hopefully mine too, but I'm guessing you'll see it. So the C4 pathway, lots of different families. It's evolved many, many times separately. So it's that kind of gives them hope. The idea that lots of different plants have played with this and they've been able to evolve this, that perhaps they can get it to happen. Okay, the other group of plants that I showed you in that very first image earlier, let me go back, is the cannon cycle, or the cannon pathway. Now you're looking at a beautiful desert environment where we're taking what the C4 plants experience to the extreme, right? You have really hot, consistently hot conditions during the day, very drought prone year round. And I mean, yes, they do have monsoon season, but it's very ephemeral. It's a huge downpour, and then that's it for a while. And so these plants have some drastic environmental conditions to adapt to. And so you can see these giant saguaro cacti are very happy. You got a thriving ecosystem there. So what are they doing? And so when they looked at these plants, they could see that they do have special adaptation. They were first discovered in the Crassulaceae family. So that's why they named it CAM. It's short for Crassulation acid catalyst. Unfortunately, they named it too soon because now we know non-Crassulaceae family plants can also do this. but the name had already stuck. So that's the origin of it. So yes, a lot of succulent plants will do CAM, but not, there's other non-succulent plants as well. And remember, every plant has a C3. It's just that this CAM cycle is kind of tacked on top of it. And what it allows for is it really helps these plants deal with conserving water. So they have a greatly reduced water loss. And they also accomplish this idea of concentrating CO2 around Rubisco. So they kind of take it, they up it a notch with the idea that we're really good at conserving water, more so than C4 plants. C4 plants are better than C3 at conserving water, but CAM left them in the dust. CAM plants really know how to conserve water. So the way that they do this is instead of a spatial segregation, like what we saw with C4, they do a temporal or a time segregation. So what that means, they do certain things during the night and certain things during the day. And so it's a temporal. So if you're a giant saguaro cactus in the beautiful deserts of the Southwest, and it's high noon, sun's shining on you, and you're like, yeah, I want to do photosynthesis, but boy, there's no water there. Would you want to open your stomata to start photosynthesis? No, that would be death pretty quickly. And so what they do is they save that activity for the night. So if you've ever camped in the desert at night, you know that the temperatures can plummet compared to day temperatures, and you can usually feel a little increase in humidity. So both of those conditions, increased humidity, cooler temps, that would be the smart move. Go ahead and open your stomata under those conditions and take in CO2 at night. But the problem is you can't run photosynthesis at night, including the Calvin cycle. The C3 cycle, can't do it at night because some of those enzymes need light. So then the plant's like, okay, I'm going to open my stomata during the night because that way I won't kill myself by losing too much water. I'll take in CO2, but I can't act on it right away, so I got to store it. And so all night long, they're stuffing CO2 essentially into the vacuole of their cells in the leaf or the stem tissue, whatever part of the body's doing photosynthesis. Then during the day, they're going to have the sun rise. They're going to go, okay, I'm going to shut down my stomata so I don't dry out. And then they can take all that stored CO2 and push it into photosynthesis and the Calvin. So fixation is talking about the idea of running the C3 cycle, taking that CO2 and fixing it into carbohydrates. So it's basically the same players that we saw in the C4 cycle, at least most of them. It has a lot of the same molecules, so that's nice. So here, this is both day and night. The night is on the left and the day on the right. You can see that actually this is happening in a single cell. So it's not like they separate a bundle sheath versus a mesophyll. They don't do that. So let's focus in on what's happening at night. So this is the night portion of that previous slide. So at night, go ahead, open up your stomata and bring in the CO2. So then what you see happening is it's the same enzyme facilitating this reaction. You have PEP carboxylase, which they abbreviate down to pepcase. So pepcase is sitting there, it'll take phosphoenolpyruvate into its active site, CO2 comes in, reacts those two together, and you end up with oxaloacetate again. And then that gets converted to malate. And then malate has carbon dioxide now as part of its body, part of its chemical structure, and they stuff that into the vacuole on there. So another word for malate is malic acid. It's the same thing. ATE means it's an acid when you see ATE on the end of the word. So you can measure the pH of the vacuole and see it getting more and more acidic. as all that malic acid is getting dumped in there. Okay, so in words, this is what we're doing. So by the way, that HCO2, that's how they depict CO2 once it's dissolved. That's how it shows up as HCO3 minus. So don't get confused by that. That's still CO2. It's just been dissolved. So PEP carboxylase. Oh, one thing about PEP carboxylase you should add into your notes. That enzyme is so picky. It only will take in carbon dioxide. O2 is not allowed. It does not fit anywhere into an active site. So this is a great enzyme to have CO2 interact with right away. As soon as it comes in, if it can get into the PEP cases and active site, that's great because hopefully PEP will be right there and they can just run this really, really efficient. So get that malate, stuff it into the vacuole all night long. Then let's think about the sunrise. The sun starts to beam on the saguaro cactus, then what? Well, then you have to think about, okay, we want that CO2 around Rubisco, so we need to decarboxylate the malate. So you can see here they're showing the vacuole, the malate leaving the vacuole, getting into the cytoplasm, and then the CO2 gets cleaved off and fed to the C3 cycle, which is happening in the chloroplast. But it's all in the same cell here. This is all happening in the same cell. So this decarboxylation, that means you're taking the CO2 off the malate. So that's great because then the CO2 gets to the Calvin cycle, the C3 cycle. Rubisco sees it. And so if you see this, if you think about this happening over and over and over, that means you get a lot of CO2 around the Rubisco. And then the other product, again, is pyruvate, just like what we saw with the C4. And so a little different here. So what they do here, instead of regenerating it right away back to phosphoenolpyruvate, what they do, you see this arrow, arrow, arrow. What they're trying to show is, yeah, it's a complicated, long pathway, but eventually you get starch. And so they just stuff it into starch until nighttime. And then at night, what they do is they mobilize that starch. So you see starch getting mobilized, arrow, arrow. So what they're trying to show here is that not really mobilized, broken down is a better word I think. It starts to get broken down at night to create the phosphoenolpyruvate. And then during the day, you have the pyruvate getting converted back to starch. So really quite a slick invention through the process of natural selection. You have this evolution of this CAM pathway, and it greatly enhances these plants to not just survive, but to really nicely adapt and even thrive in really what we would consider very hostile conditions. You know, desert conditions, long stretches of drought. What they found is pretty cool. Some plants, so far we've only discovered just a few, but some plants are what we call facultative, these CAM plants. So this is really interesting because it shows their flexibility on this CAM pathway. So if a monsoon sweeps through the desert and they get deluged with just this gorgeous water, then it would make sense, if you could, to turn off the CAM pathway and like just put that on hold. Let's just run the C3. And that's what they do. There's a handful of CAM plants. So facultative is the word we give to that kind of situation where we say they can run it or not run it if the conditions are right. And so lots and lots of water, some of these CAM plants can shut down this before because it's an extra cost. And, you know, it's just extra work. And so shut it down. And then during the monsoon season, just run the C3. But then, not too long after that, you're back to drought. And then they can turn back the CAM. So scientists are super interested in figuring this out. Like, how do they regulate that so well? How do they make that switch? Because again, this idea of can we create CAM plants that aren't, plants that CAM, put that in here, these plants. So they're looking at this idea. Obviously, going to drought season, CAM should kick in. So they've looked for, okay, who can do this? And they're finding, again, it's lots of different families. It started out, initially, they discovered it in Cassulaceae. That's why it's named CAM. And so Crassula genus, Kalanchosedum, those are many, many species within those genera. But lots of other, well, 38 other, well, that's probably more than that, but 38 different families can do that. So that's a lot of different families of angiosperms. And then if you look at a family of plants, like we're not saying that all of them do CAM. So some of them do CAM. And so some of them, if you look at one family of plants, some do just straight C3, some will be C4, some will be CAM. So it's kind of all mixed up on who can do these cycles. It's not like just one family or something like that. So again, with this idea, about 60 different times, this CAM pathway has evolved. So again, it's not coming from a single point in time from one event. It's lots of playing around through the evolutionary process at different times that have allowed these different families to come up with the CAM cycle. So here are some important ones commercially: cactuses, euphorbias, pineapple, vanilla. So the vanilla orchid, that's the only orchid that gives us something commercially that we use from it. Agave, what do we use from agave? Tequila, yeah. Orchids, of course, huge business. So all of these are very, very big in the business world. And you can also find CAM showing up in more primitive plants, so more primitive than the angiosperms, and even more primitive than the gymnosperms. So it's saying that, the CAM pathway, do I have that? Let me put that in here. I think it's on a later slide. Yeah. So the first thing to evolve was the C3 cycle. And then because more primitive plants can do CAM, that was the next invention through the evolutionary process, was the CAM cycle. And then eventually the angiosperms came along and said, oh yeah, well, we can be special too. And they said, just these four through the process of natural selection and evolution. So, and again, here's some comparisons on conservation of water in their body. So CAM plants definitely don't lose very much water when you compare them. Look at the difference: 400 to 500 grams of water lost per gram of CO2 coming in for a C3, and it's super reduced for CAM. And then C4 is kind of so. I just want to say a little bit more about the CAM cycle and like some of the applications that I'm trying to do with this in terms of climate change and just producing more commercial crop. But we'll say that because we're out of time. So we'll get into that on Wednesday.